The genomics core is almost 30 years old, which has been in operation longer than almost all the other core facilities in the School of Medicine. But over the years we've evolved, and now we really provide more comprehensive support to basic and cutting-edge genomic services. So those services would include Sanger-based DNA sequencing, genotyping, gene expression profiling, and gene panel sequencing using the latest next-generation sequencing technologies.
This particular item is the PGM, and what that allows us to do is do some very high throughput DNA sequencing. For example, this is one of the chips here, and this particular chip has 6 million wells, so it allows us to sequence up to 6 million individual DNA fragments at one time and then piece that back together using a software algorithm.
We have the ability to take and not only do the discovery part, but now move that right into implementation, into the clinic. Very few academic institutions can do that.
We're delighted to have the core on campus. We've made extensive use of the core to do discovery-based experiments. So our main focus is in the area of acute and chronic pain, and we're particularly interested in pain caused by chemotherapeutic agents used to treat patients with cancer. We've been really interested in discovering sets of genes or signature pathways that are either evoked or depressed or repressed when the chemotherapeutic agents are given. So we've used the Genomics Lab to look for novel sets of genes, pathways, either known or novel, that we can then think about targeting with future drug therapies.
One of the more recent discoveries that we've made are genetic factors that influence response to a commonly used drug called clopidogrel, also called Plavix. This is a medication that's used very commonly in people who have had stents placed in their hearts because of coronary artery disease. And as it turns out, about a third of people have a genetic variation that actually increases their risk of not responding to clopidogrel therapy and therefore increased risk for developing recurrent heart attacks. We've been able to make that discovery through the work that we've been doing with the genomics core. And now, in addition, we've been able to extend those findings to actually implement CYP2C19 genetic testing.
The Translational Genomics Laboratory is a CLIA certified, so that's the Clinical Laboratory Improvement Amendments Act that governs laboratories that perform genetic testing or any testing actually done in a laboratory for diagnosis, prevention, or management of conditions or diseases. It's unique to this environment, I think, that we have the research capability of doing things like with CYP2C19, the GWAS studies that resulted in identifying variants in CYP2C19 having an effect on clopidogrel efficacy, and then be able to bring that now to implementation studies in the CLIA laboratory with results going back to the clinicians, and being able to follow through how we can bring that research finding into actual clinical application.
Our plan is to continue to evolve and provide the latest genomic technologies to our investigators. And I welcome investigators to come and talk with us, tell us about their research projects so we can figure out how we can advance their research.